Yeah, so thank you so much for having me today as your speaker. It takes a lot to come out on a Saturday to be learning about some pretty intense science, I think, but I hope it will be worthwhile for you as well. Yeah, so just to preface, I am a graduate student at Caltech, but I also am a graduate student at the National Institutes of Health, which is in Washington. And it's sort of what I work is between going back and forth between Washington, DC and California in order to try to develop sort of new therapeutics against targeting Parkinson's disease and other neurological conditions. And I think today's talk, like I am going to go into actually what we're doing or what is the project mission, but I think more importantly, and what I hope to focus on today is really to understand, well, what is this disease and because it's unsolved right now. So why is it unsolved? And what can you do as the youth of tomorrow, basically to get towards this goal of actually helping patients? And how can we use science to solve these problems? And I think that this is sort of a call to action for you. And so I'd like to start off. So when I was an undergraduate, there used to be this sign on one of the buildings that asked, why are things, this is a quote by Johannes Kepler, who is an astrophysicist, basically, who discovered why the planets move in their circular orbits. And he asked, why are things the way they are and not otherwise? And so whenever you think about the way things are in the world, the question is, there are these paths and laws that dictate why things occur and why things not occur. And so if you can get into the roots of the mechanisms that drive these things to happen, this is the way we can make the biggest impact, is to figure out what these things are, and then from these ideas, try to modify them, I think. So I'd like to sort of start out today, mostly actually by talking how I got into science, and maybe hopefully this helps others maybe to think about if science is something you're interested in, how you can actually make a difference. And I think that there is basically opportunity for anyone, especially young students, to get involved and actually make a difference starting from today, basically. So my talk, though, I'll talk a lot about the background behind Parkinson's disease and the pathophysiology that probably leads to it. And today's talk, I'm really going to be focusing on my research, which is looking at the mitochondria, which is a piece of the cell that's really important for not only keeping the cell alive, but it also dictates when the cell dies, and why we think mitochondrial dysfunction is one of the causes of Parkinson's diseases, and why does this lead to what I and other people are doing to try to target it in order to rectify the disease. Some things I sort of want you guys to think about is, like, when we talk about the burden of a disease, what is really the true cost of the disease? Because it's not only the symptoms that are one person, it also affects their family and their community. And so really the tally is not just what's going on in one person's brain, it's a lot more than that. And we need to think about this when we try to treat disease, because it's a whole community and really a country and world effort. And I think this is sort of one of the missions maybe of your group, to think about how we can help people best, because it's not only people who are affected, it's their family and everything associated with that. And then how do we actually figure out what causes a disease when we really have no idea? So we think of problems like, let's say, COVID. We know what causes COVID, right? And actually, the world has made a lot of amazing strides towards treating something like COVID. But for Parkinson's disease, we've known about this disease for a long time, and we still don't have a cure for it. So why is that the case? And how can we maybe get at that? And why potentially maybe will we not ever have one perfect treatment? And that's what really makes solving something like Parkinson's disease very challenging. So I hope that these things will be more clear by the end of the talk. So I'll start by talking about how I actually got into science, because it's sort of, I'm still sort of early, and I think it maybe provides an opportunity for you guys to think about how you could get involved if it's something you want to do. So I'm originally from New York. I'm from Long Island, which is the suburbs that are just outside. Let me get my just outside of New York City. So in some ways, it's kind of similar to Southern California. There's big cities, and then there's a lot of areas outside where people live, like the Inland Empire and such. So I'm from over here. And I think part of the reason I really started to get into molecular biology and science is because there's actually a very famous laboratory on Long Island called Cold Spring Harbor. You might have heard of it in like a biology class, and the reason is because this is where the structure of DNA was solved. And they do a lot of really good outreach, and this is what got me involved, because in one summer there was a sort of a camp for potential high school students who were interested in molecular biology, and got a little bit of a hands-on approach to seeing what was going on in the lab. And that really inspired me to try to get into this field. I think a concept in science is all about mentorship, actually. So you can never learn, like you can learn as much as you want on your own, but really it's this learning from other scientists that actually teach you how to be a good scientist, probably. And I am definitely no different. I've had extremely good mentors throughout my career who have got me basically where I am. So it's all because of respect for basically these elders in science that have enabled me to be where I am today. So the first, and I'd like to honor them here. So Dr. Ted Brummel was a scientist basically at a local university on Long Island, and he let me come to the lab when I was a high school student, basically. And I think importantly, I sort of didn't get much done, really, but I still had the opportunity to think about what science is. And I think that's very different than the science you might be learning in a class in high school, which is a lot more about learning facts and things. But what science really is how do we find something new? And how do we take the things we know, which you're learning a lot of in school, and then figure out what's next? Because there's so many problems that need to be solved. And we'll never run out of science to do. So don't worry, it's not all done. And then from there, because I worked in this lab, I was able to get into a university that was quite good. And this was a small school in upstate New York. But there too, I continued to work in the lab, and I had another mentor who let me start immediately when I got to my undergraduate, and that was Dr. Engda Hagos. And Engda is also an inspiration because he was a refugee from Ethiopia and came to the US and basically got into science, and he's really a role model for a lot of community, especially people from Africa, and there's not that many scientists who have this journey. So he's really inspirational, not only to me, but all of my peers, I think. And so that allowed me to continue. And that's actually where I started working on mitochondria, which I'll talk about a lot today. And from there, I worked at the National Institutes of Health. That is this government body that's outside of DC, and they fund a lot of the research that you hear about. You probably hear about them now because of the COVID pandemic, and a lot of stuff was going on there. And I was at the NIH when COVID basically happened, and it was sort of a wild time because everybody started working on COVID, and Donald Trump came to the NIH campus, and it was a whole mess. And there I had another really great mentor who was Dr. Tatiana Rostatseva, also has a really cool story. She was born in the Soviet Union and then got into science because of her father, but basically escaped the Soviet Union before it collapsed and ended up in the UK and then in the US, and then has continued her career in science as a scientist at the NIH. So can I answer any questions before we continue?
Well, if you guys have any questions, just feel free to either type in the chat or
No,
you can.
this is should be more of a discussion.
Yeah, discussion.
It's really good. I can talk forever. You will probably notice. It's better. You probably learn more by having the discussion.
Discussion. Yeah.
I have a question.
Yeah.
Why did you choose Caltech, so far away from New York?
Yeah, so I came here. I'll sort of explain that. So I came to Caltech, and I think Dr. Cho already gave a talk to you guys potentially, who also is from Taiwan. But I actually came here to work with Dr. Cho because I had interest in sort of developing a new therapeutic for Parkinson's diseases. And I was at the NIH at the time, and I met another scientist, Richard Ewell, who's very big in this field. He's sort of an old famous scientist, I guess. And Dr. Cho is more new, young, and doing really cool stuff. And she basically allowed me to come to Caltech, do the project I wanted to do in collaboration with the NIH. And so here I am. And yeah, it is far away from home. And that is really probably the hardest part, actually, because family is really extremely important to me. And yeah, unfortunately, I am not in New York where everybody is, but I am going back and forth pretty regularly. And I guess more generally, why I picked sort of the research field. You guys, if I'm not mistaken, this is sort of like Buddhist influenced philosophy, yeah. So I think that is also very important for me, is finding the correct path in life that doesn't lead to extremes and try to do good, essentially. And for me, research was basically this sort of center path, perhaps, because it's exciting, it's really new, but at the end of the day, you're actually doing something that helps people. And one of their goals in life and in this world are hopefully to help our fellow people around us. And so I felt like this was a great path, because if you discover some disease causing thing, you can really help a lot of people at once. And the power of our world right now is we're very connected. And so something small you do in the lab can potentially help the whole world, which feels really good. So this felt like a good way to make a huge difference while also having a good time, because science can be a lot of fun as well. And the people who work in science tend to be very interesting. And this can be really a good environment. But yeah, so this is sort of why I think I decided to go into the biomedical research rather than something else. So today, yeah, I'm gonna hopefully tell you now about the diseases itself. And Parkinson's disease, what you need to think about is that because we don't know necessarily exactly what causes the disease, what we're looking for are the symptoms. And it's really the symptoms of a disease that tells you you probably have this condition. Whereas like COVID, for instance, the difference is you have COVID because you have this virus. And by definition, if you have this virus, you have COVID. But Parkinson's disease is not like that, because we don't know what necessarily the direct cause of it is, and it might vary between patient to patient. And that's hopefully what you'll learn a little bit about today. And so what we're really looking for in patients are, do they have these sort of phenotypes or symptoms, basically? And the most common or most noticeable, does anyone, or let me ask this question, has anyone not met anyone or is affected by anyone with Parkinson's disease? You can raise your hand. So it seems like everybody might be associated or knows somebody who does have this disease. And I'm sorry to hear that. But yeah, unfortunately is the second most common neurological condition in the US. So if you've met someone, what you know is that what they are often having are these tremors, and it makes it really hard to do basic things like eating or holding things. And these affect daily life. They have trouble often walking, and their walking is sort of stilted. So they often start walking, they either cannot stop walking, or they basically get paused. They sometimes have trouble speaking, and they also have rigid face. So it's hard to make facial expressions. And this is called bradykinesia. And these are all the things that you see going on in the patient just watching them. But there's actually a lot more going on that happens within the patient's mind and in their sense of well-being. And I think it's important when if any of you become doctors or associate with people who have these diseases, it's not only what you can see on the outside, there's a lot more that's going on. And Parkinson's disease is also associated with increased depression and anxiety and trouble sleeping. And so these things really wear on the patient, not only what you can see from the outside, but there's a lot more that is probably very challenging for them that we don't see. At really late stages of Parkinson's disease, what can happen is you also start getting dementia, and dementia is the loss of memory and other things. So this is in very late stages, this will start to occur. So now what I want to sort of think about is what is actually one of the, why is this not going? Sort of what is actually the cost? And I picked this slide, which is in dollar amounts, but really it's not necessarily dollar. It's just one way to quantify what is the cost of a disease. And it's not only the patients themselves, of course. It's really these patients, they lose out because they can't continue, let's say, normal life. So if you're 50 and 60 and you still have years of working or doing what you like, what Parkinson's disease is going to do is basically prevent you from living the type of life that you'd like to lead. And this all has a cost associated with it. The cost of caretakers who provide care for the patients is extremely high, because over time the disease gets worse and worse. And typically people actually do not die from Parkinson's. What really is happening is that their lives are becoming more and more difficult over time. And this is usually over the course of 10 years. And then it makes it more likely for them to die of something else. So it's really a wear and tear. And this is why it's so painful for the families and the people with it, because of this long pronounced decline. So if we can hopefully slow down this decline or treat it in any way, would really have a lot of societal benefit. And we know that in the US and other countries, the average age of people is going up, and this disease is associated with aging, right? And so as people age, or the population ages, we're going to have more and more of people coming down with Parkinson's disease, unfortunately. So this is a really big need as this becomes a higher and higher percent of the population who has this disease. So it's definitely a huge unmet problem. And this is why we're working very hard, hopefully, to find new cures or treatments for it. My slides are not going. Okay, so what is actually going on in the brains of people with Parkinson's disease? We know Parkinson's is basically this collection of symptoms, but why do all these people have the same symptoms? And this is because parts of your brain are doing very particular things in some cases. And what happens with Parkinson's disease is that a special part of your brain is actually directly affected. And if this part of your brain, which is known as substantia nigra, starts to not function properly, this is what leads to the symptoms. And can anyone tell me, I guess, like what, does anyone answer sort of, does anyone know what a neuron is? Anyone can answer. No wrong.
Just a cell that passes on neural signals.
Yeah, so these are the cells that make up our brain. They're very specialized because they basically take a signal and then they send the signal outward. And they do that. Does anyone know how they send the signal to the next cell?
Neurotransmitters.
Yeah, exactly, neurotransmitters. And what's special about the neurons in the substantia nigra is that they produce the neurotransmitter known as dopamine. So they're producing a lot of dopamine for the rest of the brain, essentially, and they're delivering it and activating other circuits. So brains, a lot of the way they work is that one set of neurons, they do something, they work in conjunction with each other, and then they send a signal to the next part. And it's like in a computer, this processor sends a signal to this next, and this does some other job. And this is how the brain generally works. So what these neurons do is they produce dopamine, and they can activate or deactivate other parts of the brain. And so what happens in Parkinson's, what's happening is basically this group of neurons that are all producing this dopamine start to die. And what this leads to is the type of symptoms you see in Parkinson's. So the question really is actually, we know that it's definitely the death of these neurons first that cause the gating and the things you see in Parkinson's, but it can actually continue beyond the cell type or start potentially somewhere else. But the symptoms really start because these cells start to die. So currently, there's no really good way to treat Parkinson's. We'll talk about this. The most common way is basically to provide external dopamine, because basically the brain is not producing enough of this because these neurons die. So how can we fix that, at least to begin with, is by adding dopamine. However, it's not exactly like that, because dopamine, it is a neurotransmitter, but it actually cannot pass from the blood to the brain. This is known as the blood-brain barrier. So in order to get around that, what people learned in the 60s was that they could do a modified dopamine. So it's very similar to dopamine, but it's chemically modified. This is called L-DOPA, and it can get into the brain then it gets processed and becomes dopamine, and then it can fulfill the role of the lost cells that died during the Parkinson's disease. The problem though, and this happens with a lot of medicine, is that you add more and more of this drug and the brain becomes less and less responsive as more and more neurons die. And so it stops being very useful later in treatment. So people also give other drugs that basically so the tremors, so you can stop all the tremors by adding anticholinergics. And what these do is basically stop all the muscle movement and calm that down. So these are basically treating the symptoms of Parkinson's disease. So these things that you see in the patients, but not what's actually causing the death of those neurons. And I think the big thing for the field right now, and I still think it's not totally known, is what is causing these neurons to die and how can we fix that? So this is called a disease modifying therapy, something that will actually treat what is the true cause, not just the symptoms. So today I'm going to tell you about what I'm really passionate about, which are a part of the cell known as the mitochondria. And I guess this is sort of a meme now. So tell me, what does the mitochondria do?
The mitochondria is the powerhouse of the cell.
Yay. And I'm going to tell you it's more than that today. But I think mitochondria are really cool. They're inspiring because there's this complex structure that's within the cell. So what I'm showing here on the left are a group of cells that have been stained such that different organelles, and does everyone, so an organelle, basically a cell is composed of these structures, which are like organs in the body. A cell has very tiny organs, so they're tiny organs, organelles in French. And what I'm showing here are these cells that are stained. This is the nucleus, so this is where the genetic information of a cell would be stored. And then in purple, these are filaments of actin, and this is the skeleton of the cell, the cytoskeleton. And then in green, what I'm showing are the mitochondria. And I think when you have heard about them, you probably think they're like a bean, something like this. But actually, mitochondria take all different shapes and sizes. And I don't have a video here. I probably have somewhere on my computer, but actually they're very dynamic. So these beans and strings are actually moving about like spider webs, flowing through the cell at all times. And the cool thing about mitochondria is that they form these networks, so the beans can connect together and they can break apart. And there's a lot really going on with this process that keeps them functioning properly. But if we look closer at every mitochondria, they're composed of a few different parts. And the mitochondria, it comes from this Greek, which is chondria, I think is silk, and mito is like compartment. And it sort of looks like a bunch of, if you basically stuffed a tube with string or something. And that's kind of what they look like. This is an electron microscope image, so it's a higher resolution, so you can see more detail within the cell. And then what you see is that the mitochondria is composed of three major parts. There's an outer membrane, and this is what connects the mitochondria to the rest of the cell. Then there's a space between, and then there's a second membrane, which is really, really convoluted. And this is known as the inner membrane, and these convoluted networks are known as cristae. Now, within this cristae, which is basically a membrane within a membrane, is the matrix. And the matrix of the mitochondria, I don't know, does anyone know what they also have that every cell has? Anything you know is good, so please.
DNA.
Yeah, so they have their own set of DNA, which is very special. And does anyone know why we think that mitochondria, unlike other organelles, have their own DNA? Where did they come from potentially?
It was believed that they were prokaryotes, and then it got followed by a cell, and so.
Yeah, that's exactly right. We believe, and I think it's not that we believe, this is almost 100% true, that mitochondria were once free living bacteria, basically, that entered a bigger cell and then basically created what is known as symbiosis. So they produce energy for the cell, but the cell, and they live within the cell as well. But now they're not their own free living. So you can't take mitochondria now and take them out of the cell, they will live for a little bit, but then they will die. So they're not free living anymore. They're entirely one part of the cell. And even though they have their own DNA, that DNA does not actually contain most of the proteins. So everyone, we know that every gene can become an mRNA that can become a protein. The mitochondrial genome is actually very small. It only codes for 13 proteins. And there's thousands of proteins that actually make up the mitochondria. So most of the genes that maybe were once in that bacteria they've sort of moved out and they've entered the nucleus. The cell makes the proteins for the mitochondria and then sends them there. Only some of the genes still exist within the mitochondria's own genome. So what is the mitochondria actually doing? So I think this is what's taught most commonly, of course, is that they're producing ATP, which is entirely true. And the way that this happens is that each mitochondria has a ton of different proteins within it. And these do a very complicated job of turning the basically food products that you eat into usable chemical energy. And this is known as ATP. And the way that this works is essentially that electrons are transported through different protein complexes, and what they're ultimately, the energy from each of these electrons going from a higher potential energy to a lower is basically used to pump hydrogen ions, which is just a proton, into this inner membrane space. So sort of pop question, does anyone know what will have the higher pH, the inner membrane or the matrix? Not you, Edmund. You have
Inner.
So which one has a higher pH? Michael?
Sorry, inner.
The inner membrane space, yeah. So you're pumping protons, that increases the pH. And so the pH in here is about 1. So if the pH in here is about 7.8, the pH in the matrix ends up being a little bit more acidic. So it's 6.8. So there's about 1 pH difference between the inner membrane space and the matrix. And that gradient is actually what's used to drive the creation of ATP. So there's a really cool protein in the mitochondria in this inner membrane that turns this proton movement into the generation of ATP. It's called the ATP synthase. And this ATP then can get pumped back out into the rest of the cell and can be used for everything that the cell needs to do, basically. So that's all well and good, but it's actually not only, it's not the only thing the mitochondria is doing. It's also producing tons of other types of biomolecules that the cell needs. So I think of it a lot like a general factory. So for instance, I think this is probably that we have sex hormones like estrogen and testosterone, and they're produced originally from these molecules called cholesterol. And actually, cholesterol gets directly imported into the mitochondria. It gets modified into a series of other compounds. Those compounds get back out of the mitochondria, and then they're converted into things like testosterone. And that can be used for many other things. So it's doing a lot more than just producing energy. And the other thing, I'm not sure if this is taught in like high school biology now, is it produces life for the cell basically by producing the energy the cell needs, but it's also responsible for cell death. Does anyone know what that's called? Yeah.
Is it,
Oh, sorry, go.
sorry, is it apoptosis?
Yeah, so exactly. And do you think, I guess this is sort of a more general question, do you think apoptosis is necessarily a bad thing or a good thing, or why does the cell cause this to happen? Any ideas? This is sort of open-ended, so there's not a really wrong answer here.
To prevent the cell from overgrowing into cancer.
Yeah, that's one thing. Actually, apoptosis was discovered because if you've ever seen like a developing embryo, this embryo grows out into these massive structures, but actually some of the cells in those structures like end up dying in order for the development to occur. So when you're in the womb, actually you have webbed fingers, and the skin that's in between your fingers dies off. And this dying off is it's programmed. It's the cell intentionally undergoes this to become the fully formed organism. So it's not necessarily bad that cells die. And the reason mitochondria are extremely important is because one of the ways the cell programs this is when the mitochondria actually become damaged. So cytochrome C is this protein. It's in the inner membrane space. It's soluble. That means it's floating around. And it actually moves some of these electrons from one complex to another complex. And that's what its day job is, basically. And this is sometimes we call what a protein, if a protein has one main job and then does something else in special conditions, we call it moonlighting, because people who like work one job and then at night go do something else, it's called moonlighting. So cytochrome C is one of these proteins that moonlights, and that's because if it gets out of the mitochondria, it actually initiates this process that ends up with the cell dying. And so a lot of times this is great because this is what needs to happen for biology to work. But we also know that if mitochondria become damaged randomly, this can cause the cell basically to die as well, which might not necessarily be good. If you have a cell you don't want to die. And what is a cell we do not want to die that doesn't grow back? Anyone know?
Neurons.
Correct. Yeah, so neurons are very slow to grow and they often never grow back, especially the ones that are deep within your brain. And so if those cells end up dying, there's not really a good way to fix that. So damage to the mitochondria has the potential to really damage neurons effectively. And I think that's what we're going to go to today. So one other thing I want to talk about is something I study a lot, which is what happens if you have a damaged mitochondria, but your cell does not want to die. Well, before the cytochrome can be released, you can actually clear up this damaged mitochondria before it has the chance to kill the entire cell. And one way this happens is called autophagy. Can anyone guess what that means? It's sort of Greek. So auto means self, like a robot is automatic. It functions on its own, autonomously on its own. Auto is self. Phagy means to eat. So the cell is actually eating itself, autophagy. And this has been known actually for a long time, since the 50s, that this process was occurring in some cells, especially when cells start to starve, was that these membranes were basically forming around organelles like the mitochondria. And these are the electron micrographs. So you can actually see this process occur. And what's happening is that these pieces of the cell, like mitochondria and other parts, are going engulfed in this membrane here. And then that membrane is going to fuse around them to create what is known as an autophagosome, the self-eating vesicle. And there's two membranes here, and that's why they're very discernible if you look at an electron micrograph image. And then does anyone know what the lysosome does?
It recycles the cell's material and then repurposes it. Okay.
And you know how it does that by chance?
It eats it up. I'm not sure.
Yeah, so there's these proteins that are kept within the lysosome, and they're called proteases. And proteases are proteins that destroy other proteins, basically. So they're like the scissors of the cell. And there's not only proteases, there's nucleases. Those are things that destroy nucleic acids like DNA and mRNA. And there's tons of enzymes that are just meant to cut things into small pieces. So the lysosome is this organelle that holds these things. What it ends up doing is fusing with the autophagosome to create this autolysosome. And do you know why the lysosome is not normally active and why it doesn't destroy the cell typically? Well, so yeah, basically what happens is that these enzymes don't normally function at cellular pH, but what happens if you want this to turn on, this organelle, you start lowering the pH in it. So you pump in these hydrogen, you pump out, and then basically you're going to end up lowering the pH within this compartment. And then this will activate everything to destroy whatever's inside this. So mitochondria can actually be engulfed in these process. And this is known as mitophagy. So it's one subset of autophagy. And this hopefully will stop mitochondria from destroying the entire cell if that's not what you want to happen, like in, let's say, a neuron. Okay, so now going back to the disease, how do we know what can actually cause a disease, something like Parkinson's, and how can we maybe find clues within nature and looking at these cells to figure out what is going wrong? And if we know what's going wrong, then maybe we can figure out ways to fix it. And I think this is like important in that when you're in school, you're learning a lot of different facts, but now as you come out and if you want to get into science or medicine or anything, it's about figuring out what we don't know. And this is one of those things we really do not know, or not for sure anyway. But today I'm going to sort of bring you through sort of my logic in what I think is going wrong in at least some of these patients and how we think we might be able to treat it. And this is sort of like playing molecular detective. Okay, so one of the things that makes Parkinson's disease very complicated is that some patients are known as familial. And what that basically means is it runs in their family. So their parents, their grandparents had Parkinson's disease and they're likely to have it. And do you know why this can be passed? What might be going on in these patients, anybody?
Errors in DNA gets passed down, primarily probably an error in one of the regulation proteins.
Yeah, and so there's might be some gene that has a mutation that causes Parkinson's disease. I think the complicated thing that you need to think of is a lot of mutations in the cell will end up basically killing the cell. They cannot function. So what must happen, and so you'll never actually know that basically those eggs, when they're in the womb, they never actually develop. And this leads to a lot of premature, basically pregnancies that fail because there are a lot of mutations and they will prevent cells and the organism from ever getting developed. The thing about Parkinson's disease is people are living, right, for 50, 60 years, and then they're getting this disease, right? So it's really complicated. Whatever is going on is very subtle. And this makes it really hard because you can imagine in our cells, if we have them in the lab, whatever is going wrong is something that's going to go wrong over the course of 50 years, which we obviously do not have the time in the lab. So it makes it really hard to figure out what is the cause of these sort of aging-related neurodegenerative diseases. However, the thing is that once we do know what are the genes that are actually causing this within the family, we can get a better sense of what are those genes actually doing and potentially what is the pathways that are leading to this disease. But it's more complicated because even though some people have a family history of the disease, most Parkinson's patients are known as sporadic or idiopathic. And what that basically means is we don't know what caused their disease. So this can be because of many, many different reasons, and that includes a complex interplay between the environment. Maybe you're at risk because of a genetic mutation, but it's not obvious because it's not like everyone in your family has it. So it only is because you were exposed to something that and you're potentially at risk, or because you lived your life in a certain way that makes it more likely to get a disease. And so because of this problem, where actually most of the patients with Parkinson's probably don't necessarily have one of the obvious genes, it is very confusing to figure out what is actually causing their particular disease. And does anyone one question, I guess, is why is something like cancer hard to fix? Does anyone know? And this is sort of general.
There isn't one
because
set cause.
it can go in your blood, right?
Yes, that's why it will kill you. But why, if I figure out how to cure one person's cancer, why does it not work necessarily for everybody?
Because
There's
everyone's
like-
cells different.
But you can
Because
go.
okay, so because everyone's has different cells and like genes, so like their mutations would go on different paths. So it doesn't apply to every single person.
Yeah, and if who is the other person who's going to talk? Whatever. I just want to hear what you have to say as well.
Oh, I was gonna say like there's lots of different types of cancer. So something that cures one person's types of cancer might not cure another person's.
Right. And I think this is actually a problem for us in our language, how we talk about disease, is that cancer is one class of diseases, but each type of cancer is very, very different. And this makes it really hard to take one cancer and then try to treat it, even though it seems sort of similar. And I think one of the things we're sort of learning in the field of neurodegenerative diseases is even though people all show these phenotypes of Parkinson's disease, what is causing the death of those neurons might not be the same from patient to patient. So even if you have a therapeutic that works for somebody, it might not necessarily work for everybody. And this is probably why a lot of therapeutics have failed in this neurodegenerative disease field. And probably it takes a lot. It's just, we need to think of every single patient as a unique case. And this is sort of a push towards personalized medicine, but we also hope that subsets of patients can be treated by a similar therapeutic. And it's the same thing with cancer. People who have this certain gene mutation, which is very common, can be treated with a similar drug. And so maybe it's just stratifying patients that will help us a lot in the future. But what this does, we can sort of think about a lot of things that might be contributing to one disease. So let's focus on these familial mutations and some of the research that got into there. So I just wanted to talk, of course, we know the mutation is in the DNA. This leads to a change in this mRNA, which ultimately often it can change the mRNA itself. So you could have changes here that maybe even prevent the protein from being made, or the protein can be made with a modification that changes how it functions. So every protein generally plays a role in what is called a cellular signaling pathway, and proteins each are like little machines that are playing a role in a big network. And so you can change them very slightly and have a very different outcome, just like if you changed one circuit in a computer, one bit from a 1 to a 0, you might change the entire program. And this is sometimes how cellular pathways work, and they can go wrong because they have a slight modification in one protein. So we try to do the strongest information we can get from genes is when a bunch of genes seem to play a role in the same pathway. What that tells us then is if a bunch of proteins connect together in one circuit, and now you have patients all around the world who have different mutations in different genes that all play a role in this circuit, now we know it's probably this circuit that is actually contributing to the disease. And so this is like putting the puzzle pieces together. And this has happened over the course of many, many years as people learn about family mutations that cause Parkinson's, and then look for what are the proteins involved, and then try to piece them together, how they might be working together to create some type of function, and why does the mutations lead to dysfunction. So I'll tell you about a little bit of that with Parkinson's today. So the thing I talked about earlier was if you have a damaged mitochondria, how do I get rid of it before it kills the whole cell? And even though I said I basically showed you an overview that mitochondria can get into these autophagosomes and then be destroyed, everything that happens in your cell is orchestrated by proteins actually doing something, right? And that doesn't happen by random. It's not that they can probably randomly get into the autophagosome, but the cell needs to have a way to take damaged mitochondria and make sure they do get into the autophagosome, right? You don't want to just get rid of everything or else you don't have any energy, right? So the cell comes up or evolves ways to specifically remove damaged mitochondria. And the pathway, this is a cellular pathway I'm going to tell you about today, is called PINK1 and Parkin. We basically learned about this pathway because mutations in each of these proteins were found to cause Parkinson's disease in these families that have them. So if a mitochondria is healthy, it has a membrane potential. We talked about that before. And if it's unhealthy, before it explodes and releases the cytochrome C into the cell, it has a depolarized membrane potential. And so that means that the pH between the inner membrane space and the matrix is relatively equivalent. So not only is this membrane potential creating ATP, but the mitochondria is actually using this potential to do plenty of other things. And one of those things is actually getting proteins from the cytosol to get into the mitochondria. So not only is it the force that makes ATP, it forces proteins to get into the mitochondria. And I think I talked about this a little bit, but a lot of the proteins that are actually used to make the mitochondria are produced by nuclear genes. And so that means they're made outside the mitochondria and have to find their way in. So you can imagine that then you don't have this force that's getting proteins in. What starts to happen is this proteins get stuck on the outer mitochondrial membrane. And one of these proteins is known as PINK1. So PINK1 and another protein known as Parkin were first discovered because they were Parkinson's associated genes. So Park6 was the name of the gene because patients who had mutations in this gene were getting early onset Parkinson's. They were getting it very early. This means before 50. And there were two proteins, PINK1, which is Park6, and this other protein, Parkin. And it turns out that they actually function together. So what ends up this took many, many years to discover each step of this, but basically PINK1 gets stuck on the outside membrane of the mitochondria, but when it's stuck there, it's actually active. So this is its job. It's like a flag that tells the cell, this mitochondria is not functioning, I'm stuck here, get rid of me. And what it does is in the cytoplasm floating around is another protein called Parkin. Parkin gets stuck to PINK1, and it actually is called a feed forward loop. And this is because PINK1 activates Parkin. So this is how the cellular system works. They have these networks that feed together to do something. So PINK1 recruits Parkin, and Parkin gets onto the mitochondria, and now it's active, and it starts adding these molecules called ubiquitin to the outer mitochondrial membrane. Ubiquitin are a small protein, and they're often used to tell the cell that something needs to be done. And usually what they're telling the cell is this protein needs to be destroyed. So they're like cellular death tags. If you get tagged with ubiquitin, the cell knows to get rid of you. And if the mitochondria gets tagged with ubiquitin all over, then the cell knows to slot you into one of these autophagosomes and destroy you. And so this leads to mitophagy. And this, like all of these discoveries were basically made by one of the labs I'm working with, Sir Richard Yule, and a whole team of scientists basically discovered that these two genes were doing this really cool pathway to remove mitochondria. So yeah, I mean, won prizes for that. So there are prizes, which are not why we do it, but anyway, cool enough. Another reason we think mitochondria are involved in Parkinson's disease is not only these genetic mutations, but some environmental factors can actually cause Parkinson's diseases, and we know that they also basically damage mitochondria excessively. So there's two stories I sort of want to talk about today. One is this drug called MPTP. And this was sort of a mystery in the 70s because basically people were making synthetic opiates. So you probably should not take drugs for many reasons, but one of them is because they can have other things that are actually not what people think they are. And this is what happened in the 70s. People were illegally producing these drugs, and it ended up having a byproduct. And so patients all around the country, drug users, were coming down, young people, basically with Parkinson's disease. So they were injecting themselves and then getting Parkinson's. And what was going on there? It turns out that this compound that was accidentally being produced with these illicit drugs was getting into the neurons and then destroying the mitochondria. And so we learned, this was really the first evidence before even the genetic stuff, that damage to the mitochondria could actually cause Parkinson's. And if you gave this drug, just this drug, to monkeys, it would end up causing the death of those dopaminergic neurons that are the ones that are producing dopamine for the cell. And it's not the only one. I don't know if any of you know who Michael Fox is. He's sort of a famous actor in Back to the Future. I don't know if anyone has watched these movies, but he's very famous in the reason that actually he got Parkinson's disease very early on and became sort of a vocal advocate for funding research into Parkinson's. So there's the Michael J. Fox Foundation that funds a lot of Parkinson's disease research. And it turns out that he probably got Parkinson's disease because he was exposed to a compound called rotenone. And rotenone was used as a pesticide for a while, but we also know that it's a really bad mitochondrial toxin. It destroys the ability for mitochondria to produce energy. And when Michael Fox was a young actor, he was on a show in Canada, and they were working near downstream of a field that was basically using tons of this drug, and they were all exposed to really high amounts of this pesticide. And so Michael Fox and a lot of actors and crew from the show ended up developing Parkinson's disease. So now we know 2 drugs that end up destroying mitochondria and probably causing Parkinson's disease in some people. So the final piece of evidence that I would say that suggests that mitochondrial damage causes Parkinson's disease is that in patients who we have no idea what caused their Parkinson's disease, we can take tissue samples soon after they've died. So these are the neurons coming from their brain, and then test, how are the mitochondria functioning in those cells? And so what we've found over the course of years is that patients with Parkinson's disease tend not to produce as much ATP, and their mitochondria seem to be more dysfunctional. So at least, and those are in the neurons that are actually still within the brain. So the ones that have died are totally gone. So we know there's a lot of problems going on in the mitochondria of patients, basically, who definitely have genetic causes that cause their family to have Parkinson's, and those have been exposed to toxins, and people who we have no idea what's going on still have, it's been suggested they have a higher chance of having dysfunctional mitochondria. So now we're sort of detectives. How do we put all this information together and think about what could actually be causing a disease? And this is sort of what I'm trying to get at, and maybe for you guys to think about. It's not just because somebody tells you, it's all of these pieces of evidence suggest that maybe this is what's going wrong, and this is one thing that we can maybe test. This is our hypothesis, that mitochondrial dysfunction is causing the death of these neurons and causing Parkinson's, basically, and this is a molecular mechanism that might be responsible. So basically what I've told you today is I feel that there's 3 strong pieces of evidence for this: the toxins, the genetics, and then the association with mitochondrial dysfunction. So if we know what's causing it, how can we now design drugs and treatments that hopefully rescue this problem if we think we are causing mitochondrial dysfunction? So I'll stop, and hopefully anyone can ask a question before I continue, before it gets a little bit more in depth about what I'm actually doing.
When you're in the National Institute of Health, you stated before that you believed that there was something wrong with the mitochondria. It was called what's it called? The sub something. I'm not sure. I can't find it now. But you said that it was found in alpha-synuclein. You said that it was found in the brain and it could help affect the Parkinson's disease. Why do you believe this?
Yeah, so this is another, I sort of didn't include any of this in the talk today, but there's actually another set of evidence that there's another protein that's really involved in Parkinson's disease, and this is called alpha-synuclein. And the reason this is believed to cause Parkinson's disease is because in the patients who die of Parkinson's, what you start to see in their brain are what are these sort of plaques. They're aggregates, and they're called Lewy bodies. And if you take those plaques, basically, and look what's actually in them, it's the accumulation of this protein called alpha-synuclein. Alpha-synuclein is a small protein. It's expressed in a lot of different neurons, but it tends to accumulate not only in Parkinson's disease patients, but others who have dementia, and that's called Lewy body dementia. But we also think that this probably plays a role into mitochondrial health. So some research I did a long time ago, or a few years ago, is actually that alpha-synuclein can also get into mitochondria and prevent mitochondria from functioning. So this is sort of something that we think is tying together this association with higher alpha-synuclein that's associated with Parkinson's disease and how that could actually cause the neurons to die. And potentially we've sort of developed therapeutics that hopefully prevent alpha-synuclein from getting into the mitochondria. And that's sort of at an early stage at this point.
So was this before or after you decided that the voltage-dependent anion channel was a part of what helped the alpha-synuclein get inside the mitochondria?
Yeah, so that's another piece of research. The question is actually, well, things in the cell, they don't just go places random. They have to get into in order to get into the mitochondria, it's not simple. And that's because proteins that get into the mitochondria typically have a signal. They have a flag on them that says, bring me to the mitochondria. And then the cell does force those proteins to get into the mitochondria. The confusing part about alpha-synuclein is it doesn't have this tag, which is called a mitochondrial targeting domain. So how does it get into the mitochondria? Well, what we were working on is that there is actually another channel. So channels are proteins that allow things to pass through membranes, which are not permeable to water generally, but channels allow ions and water to pass through. And what the voltage-dependent anion channel does is allow ATP that's produced in the mitochondria to get out. But it turns out it's much bigger than just ATP. And we showed in these papers that actually alpha-synuclein can go directly through this channel, even though it's not thought to be a protein movement channel, it's for ATP, but can accidentally allow alpha-synuclein to go through. And we think this is the pathway by which this damaging protein ends up getting into mitochondria when there's too much of it. And this happens in some patients. They have what's called a triplication mutation. That means their gene makes 3 copies of itself. And so you have extra amounts of that protein in your cell. And this definitely causes Parkinson's in those patients.
Final question.
Yeah.
So what for the alpha-synuclein, besides Parkinson's, is there any other like degenerative diseases that could like also be tied into it, or is it just Parkinson's that's primarily because of the alpha-synuclein getting into the mitochondria and preventing the production of ATP?
Yeah, so there's another disease called Lewy body dementia, and this causes a different subset of symptoms, basically. So it doesn't necessarily result in the stuttering and tremors that we see in Parkinson's disease patients. It's much faster, and it actually leads to dementia, and that's because it's affecting your frontal temporal lobe, which is for higher order thinking. And those patients can also have mutations in this protein, alpha-synuclein, and it's also causing the death of those neurons. So it's potentially that these neurons are also dying in the same way. It's just that for whatever reason, these mutations cause it to be expressed in those cells rather than the dopaminergic neurons that we see that cause Parkinson's disease.
Okay, thank you.
Yeah, thanks for your questions. Yeah, so I'll sort of talk about what I'm actually working on now, which is sort of, we're trying to, even though we could possibly target maybe these pathways like alpha-synuclein, what we'd like to do at the end of the day is protect or prevent mitochondria from damaging the cell in general, no matter how they're getting damaged in the first place. So this hopefully therapeutic is aiming at not just the people who have, let's say, excess alpha-synuclein or a mutation in mitophagy. So hopefully more widely useful. And the way that this sort of idea happened is actually basically through basic research. The lab was not actually interested in creating therapeutics, and I'm going to try to go through, this is real data, so I want to sort of explain how we learn things about cell biology, and this is sort of one of those events. So the question that sort of happened is, I think I talked about basically mitochondria get tagged with this eat me signal, but why does that signal actually end up letting the mitochondria enter into this autophagosome? Well, it turns out a lot of those proteins, what happens is that after the mitochondria got these tags on them, a bunch of proteins were starting to also come onto the mitochondria. And all of these proteins basically had a piece of them that is called an FIR, and this is called a FIP200 interacting region. I didn't really talk about this before, but the autophage, you don't want to just eat every part of your cell all the time. So you really want to only start forming the autophagosome when you need it and when you have damage. Otherwise, you'll just get rid of things you actually need in the cell. So FIP200 is this protein that basically is like the signal that starts the formation of the autophagosome. And it turns out that all of these proteins that were going onto the mitochondria were basically bringing FIP200 with them. So in the lab, what we showed is that if we just forced an artificial protein that could bind to FIP200, so it sticks to FIP200 on one end, and then to the mitochondria on the other end, we could end up getting these mitochondria to go into autophagosomes no matter what happened to them before. So they don't actually need to be damaged. Anytime we add this protein to the cell, which is sort of artificial, we would force mitochondria to go through this process of mitophagy. And I'm showing this a little bit here. This is real data. This is a cell. In red, you have the mitochondria. And then if mitochondria get this purple, that means they're actually in the lysosome. So I think it was important when I said before that the lysosome is active when the pH goes down. And so this signal basically is only on when the pH in the around the mitochondria is very low. So that means they're in an autophagosome. So then what we do is basically force this fake protein to the mitochondria. And you can see that if we don't force it to mitochondria, it's everywhere in the cell. And so this is floating around. This is a nucleus, which I don't have labeled here, but it's this black hole in the middle, and it's floating around, and you have some foci maybe. But when we force it to the mitochondria, you can see that it forms this pattern, which is identical to where the red is. So now, and when you do that, you force these mitochondria to end up in the autophagosome. So what we showed in this paper is basically that if you force FIP200, this autophagy initiating factor basically to the mitochondria, you could force it to go into mitophagy without anything else happening. So even though we showed this in the lab, the question is, can we turn this into something that's actually useful as a therapeutic? So the idea here is sort of just some lingo. A ligand is just a small molecule that binds to something else. So a lot of drugs are ligands. That just means they stick like a key into a keyhole, and the protein is the keyhole. And these are small molecules. So that means that they're a chemical that we can produce outside of the cell, and then it can get into the cell and do something. So the way that this idea basically would work is that one piece of this drug would be binding to FIP200, just like the proteins normally do in the cell. The other part of this drug would bind to the mitochondria. So basically you can imagine it's like a glue. It forces this protein called FIP200 to be stuck on the mitochondria. And what that ends up doing is telling the cell to form the autophagosome around the mitochondria and lead it to be degraded via mitophagy. So we think by doing this, we can enhance the basal rate of autophagy of mitochondria in these neurons and hopefully protect against cell death. And so this is sort of how we think it would work. The small molecule compound basically sticks them two together and then ends up with mitochondria in the lysosome. And so that's sort of what I've been working on a lot in the lab. We hope that this compound that we're developing now, and it's not quite ready yet, but we're getting, we basically have one half of it done, which is we have this blue molecule found now, which we had to discover. It didn't exist. There's no known small molecule that bound to FIP200, so we had to search for one. But I don't have a lot of time today to talk about that. It's a wholly independent process. But we hope that this will basically prevent the buildup of these damaged mitochondria that happen in the cell and prevent the cell death that's associated with it. And so this is not the only way that you might think about altering this pathway. There's a lot of companies and therapeutic potential to target different parts of this pathway that ends up with the mitochondria getting engulfed in the autophagosome. There's one company that's basically working on just activating that protein PINK1 so that it's always active. And that means you have more mitochondria going through this flux. We also know that there's different parts of the cell that actually stop this from happening normally. And so you can imagine the cell doesn't want this to happen sometimes, so it has ways to prevent it. And if you prevent the thing that is preventing mitophagy, you increase mitophagy. So it's like a double negative there. So they're trying to develop small molecules that actually basically allow this to happen without going backwards in the process. And you can also, there's also known drugs that just increase the rate of autophagy generally in the cell. And so this is potentially another strategy, but it's a little bit less targeted. So I'm not the only one working on this, luckily. And we hope that in the next few years, some of these will start to enter clinical testing and seeing if this idea actually holds up in the clinic. Yeah, I think that's pretty much the whole talk I had for you today. So I'm here to answer any questions you have.
I had a question regarding Fit 200.
Yeah.
So you said that like multiple proteins will carry Fit 200 with them to attach to a mitochondria.
Yeah.
Does it only take one Fit 200 protein for a mitochondria to form an autophagosome?
That's a really good question. And the answer is basically no. What probably needs to happen is that you need multiple FIP200 on the same basically surface. And what will then happen is that FIP200, I don't have this slide here, so let me go to a different presentation so I can talk about it a little bit more. So I sort of simplified a little bit, but what FIP200 actually is a protein that already sticks to other proteins. So a lot of things in the cell are done, the jobs in the cell are done by things called complexes, and these are lots of pieces of protein coming together to do one job. So FIP200 is like the glue that holds all of these other proteins together that form the autophagosome. And the reason it's not just one molecule of FIP200 you need to induce the formation of the autophagosome is because what's actually doing this job is another protein called ULK1. And I know this gets really complicated because every pathway in the cell has a million other things going on. But what we think is going on is that when you have at least two or more FIP200 molecules close together, ULK1, which is known as a kinase, does anyone know what that means?
I think it's like a protein that helps with transduction.
Yeah, so kinases, what they do is phosphorylate. Has anyone heard the term phosphorylate? So it's just basically they can activate other proteins. And FIP200 carries with it this other protein called ULK1, which is this kinase. And if you have multiple ULK1 molecules near each other, they basically activate each other. And this causes this feed forward loop that now, if you have a bunch of active ULK1s at this site, it can activate everything else that's needed to form this membrane at that site. So what we probably just need to happen is have a few of these molecules at a site so they can start to auto-activate. But yeah, that is a really good, insightful question, and it gets into exactly which I hope everything in the cell has to happen. And it doesn't just happen because we say it happens because molecular events are actually taking place that turn a signal into something the cell can actually do. And I think that's really important to think about. It's not just these diagrams of these circles. Each protein actually has to physically do something to turn on the next signal that the cell can respond to.
Oh, I have another question now that you mentioned that.
Yeah.
If it's in a feed forward loop, does it ever become to a point where kind of like cancer cells, because like they become cancer cells because they're dividing so continuously and there's no stop? Is there ever a point where it becomes like that, where it's too much activation and there's like no homeostasis type regulation for it?
Yeah, there is a problem, and we're very mindful of this, which is that if you have too much autophagy, you can actually cause the cell to die that way because it just clears out everything it needs to survive. And this is called an autophagic cell death. So there's a quote basically that every medicine is a toxin. The only thing that distinguishes the two is the dose. And so something like the therapeutic we're creating if you probably took it at too high amounts and induced too much mitophagy, it would actually be bad because you would clear out all the mitochondria, not just the unhealthy ones. And this could kill the cell in a different way. However, it's not only that, autophagy is probably good and protects against neurodegenerative disease, but we also know it can help cells avoid cell death due to stress like starvation and other drugs that might cause toxicity in the cell. You can imagine which cells really want to survive. That we don't want to survive. What is a condition where we have cells that we want to die, but they don't want to die?
Like cancer?
Exactly. So some
Yeah.
cancers actually upregulate autophagy, and this helps them to survive events of stress, or like we try to chemotherapeutics, and they can actually prevent themselves from dying by continuing to upregulate autophagy. And so this is always this thing in biology, which is treating one disease can often exacerbate another. And we have to be mindful of this because this is called an off-target effect. And some people are working on autophagy inhibitors in order to treat certain forms of cancer that are drug resistant.
Oh, I have a question. So besides that, what Melanie just mentioned, like continuously inducing like autophagy, but like what other off-target effects does TEP200 have when you're like having this when you're trying to go through this process?
Yeah, so we don't exactly know yet what this drug will do, and we're working on that to figure out what are the things to look out for. I'm also investigating sort of separately what is FIP200 doing normally in the cell that's not related to autophagy. We think it also regulates mRNA, which is the stuff basically that's coming out of the nucleus to translate into a protein. We think that FIP200 can also help regulate those mRNAs. So we wonder whether forcing it into the autophagy pathway will disrupt something with the translation regulation. So these are sort of questions we're still focused on now.
So besides biomedical medicine, like the symptom is genetics. So do you think that maybe like the mothers, when the kid is in the womb, like maybe Michael J. Fox, besides being exposed to something in his lifetime, do you think others can be exposed to something in the womb, like because the mothers were doing something, eating something, exposed to something that can later develop to the Parkinson's symptoms of that child later on?
Yeah, I think this is definitely theoretically possible, that something that you were exposed to in the womb could cause disease later on. These things are really hard to test because you need to find what are you looking for, exposure to a toxin in some population, and then track these patients over the course of, let's say, 60, 70, 80 years. So sometimes it's hard to figure out these associations, which is a bit scary. We know exposure to certain toxins in the womb does cause disease. This is very obvious with alcohol. So pregnant women should not drink alcohol, and this is because it leads to basically developmental problems in the childs that are born. But also there's other famous cases, like for instance, I talked a lot about ubiquitin, a little bit, not a lot, but ubiquitin is this thing that tags proteins so that they get destroyed. There was once a drug in the 50s, and this was called thalidomide. Thalidomide was basically given to pregnant women to help them with morning sickness, but it ended up causing horrible birth defects in some of those children because it accidentally basically modified this ubiquitin pathway. And this actually helped us to learn about what this thing was doing because we had something that impaired it. And now derivatives of thalidomide are actually anti-cancer drugs. But yeah, I don't know, to answer your question. So far, I'm not sure of any convincing links between prenatal exposure of something that leads to Parkinson's disease later on, but it doesn't mean it doesn't exist. It's just very hard to test for.
In the same vein as that, how early could you tell if someone is at risk of Parkinson's because of family history? Because is it only when they start to exhibit symptoms that you might be able to see if there's a genetic linkage? Or
So, and let me also preface, I'm not a doctor, so I can't give actual advice, but what I would say is basically this: if someone in your family, and especially if more than one people in your family have come down with Parkinson's disease, it's a good idea to both have them genetically tested and you genetically tested, probably, to look for some of these known causative factors. And the problem with Parkinson's disease is that what we're looking for, and this is another active area of research, is can we detect the disease before it ends up with the symptoms that you see? And this will probably improve treatment. Imagine if I have a drug I know slows down the disease. However, I need to treat patients before they get it too badly, before those neurons actually die. And this is one of the things people are working on. We're doing this in the lab, which is looking for what are called biomarkers of the disease. That you could detect before someone actually develops the disease. So this is called prodromal, which is before symptoms. And people are definitely working on that for Parkinson's disease. I do not think there's a super good or well-validated test yet for that, but people are definitely working on it. And I think within our lifetimes, we will see that, and it will probably benefit us.
Oh, if you guys have any questions, you can just let Will know, and then we can continue our discussion. Or I think I have a question, but it's not entirely related to the research topic. But how did you want to focus, or how did you get into this project, or what's your how did you get into doing Parkinson's disease project?
Yeah, so actually I was more necessarily interested in mitochondria and when mitochondria go wrong. And the clearest disease that this contributes to is Parkinson's, in my opinion. So I had thought basically, when I was an undergrad, I was working on mitochondria and related to this pathway of mitophagy. And I was sort of actually annoyed in a sense. Why have we not, if we know this pathway exists, why have we not drugged it? And sort of like, this is clearly something that has not been done effectively. So then I think I thought about a lot of ways that we could improve mitochondrial health. And one of them, I was inspired, I think, by my friend. Has anyone heard of CAR T cells or immunotherapy? So this is in mostly cancer therapy. What people will do is basically engineer your body's own immune system to directly target cancer cells. So you can actually take your immune cells out of your body. You add like an artificial protein to them that tells them to go attack this cancer, and they do. And this is actually curing a lot of childhood leukemias that were devastating before and now are actually treatable. And so I was really inspired by his research, because what he's doing is effectively engineering cells to go do another new function to prevent disease. And I was working in autophagy and thought, why can we not engineer cells to attack things within the cell that are causing damage? And so can we think about ways to engineer this in a similar way? He's the whole cell, I'm just a part of the cell, but this is sort of how I was inspired to get into this project.
Do anyone have any questions for Will? So about his project, about his career for now, and anything, if you want to ask, you can talk right now.
Hypothetically, if Parkinson's was cured in a year, what would you do? Like, what would your career?
Yeah, that would be great. First off, I have a lot of other sort of interests in discovering basic biology. I think one of the things to do is making drug discovery much easier because it's a really long process. And I didn't really talk about that today, but I have a lot of sort of ideas I think that will help make it faster to develop new drugs against something. Imagine you have something like COVID come out. How can we develop and get a new treatment ready to go in much faster time? Because right now it takes many years generally to get drugs made and tested. So if we can improve this process, we'd probably really improve a lot of lives more rapidly, I'd say. So yeah.
Did you do a lot of research-based activities in your undergrad that led you to like forming projects? Because I know that you did early on in high school at the lab, but was there anything that you did specifically in undergrad for this?
Yeah, I'd say like this is semi-untraditional, but I was like really into research. So I spent a lot of time in my undergrad not focusing on class and focusing on the research, which ended up, I feel, more worthwhile for me because class is really great to get a fundamental understanding, but you actually learn a lot in the lab by forcing yourself to figure out what you need to learn. So I spent a lot of time in my undergrad. A lot of times colleges will offer credit for just doing research, your own independent research projects. And there's always stuff to be done in labs. So this is what I focus on. And I would encourage you to do the same if you're interested in science, because it's more worthwhile to get a foot in the lab and see what it means to actually do science rather than take the classes, which are important. But I think you can learn a lot more in one semester in a lab than you can in one semester in a class. And of course, with biology, things are always changing every day. New papers that come out and change everything we think we know about the cell. So it's a very rapidly developing field. And so some of the things you learn in class might not last another 10 years, unfortunately, but the skills you learn on how do I ask the questions, how do we know what we know, and why is it not otherwise? And these sort of basic higher level questions of how we actually do science will never change because this is philosophy really. And they can help, no matter how the world changes, we can always apply this mentality to figure out what comes next, I think. This doesn't mean don't go to class, but definitely spend time in the lab if you're interested in doing science, because this is where it happens. And also, if you're an undergrad, tons of labs would be really happy to have a dedicated undergraduate who is motivated to come in. And there's a lot of things in the lab that are just a lot of need more hands, basically. So there's a lot you can really contribute to research without necessarily knowing too much about it, just by being dedicated. And I think what I've learned, I wasn't necessarily the best in class or knew the most, but just spending a lot of dedicated time towards your craft and learning how to do techniques ends up being really useful, because this is often what we lack in the lab, is just the time and the hands to actually get things done. So you can really contribute a lot to research just by being committed to it.